human skin cells affect building health, issues, and solutions. We will begin in just a couple minutes here, just waiting for everyone who wants to join this so we can go ahead and get started. Thank you. Thank you, everybody, for joining us. This webinar is How Human Skin Cells Affect Building Health, Issues, and Solutions. My name is Ronnie. I am the Quality Assurance Manager of Environmental Diagnostics Laboratory, and I have with me Dr. Rajiv Sahai. He is the Laboratory Director of Environmental Diagnostics Laboratory. So we are just going to go ahead and get started with this. And so we'll begin with the main topic of this webinar, human skin cells. Dr. Sahai, can you elaborate, a little bit on what human skin cells are? We all have skin here. We all, experience it, but what makes it what it is? Let's start with the foundation on it.
Yes, very good. Good afternoon to all our participants. And Ronnie, yes, first we need to introduce the topic. And human skin cell, as the name indicates itself, that it comes from mostly the skin of human being, and there is no way that at this time with a limited microscopy we can distinguish between the human skin versus the skin of other pets and other animals. So I just want to make sure and clear that the information which we will be giving is going to be under the human skin cell itself. Well, this is very commonly observed but most ignored flora, or the dust microbiota, if I say like that. So basically, the skin is nothing but it is a protective layer of the human body, and there are three layers, as you can see in this diagram. The very superficial layer is called as epidermis, and it is followed by the layer called as dermis, and then obviously the subcutaneous layer, which is basically or mostly containing fat. So this is sort of a structure of the human skin. Some of the quality of the skin and usefulness of the skin to the body, that it is a protective layer, or it forms the protective layer of the human body, and it is also insulate our body for external forces or external causes, and it is also waterproof. So what happened when the skin cell is dead, then it become a part of dust microflora, like I was earlier telling, because every human being, or for that matter other animals or the pets having a skin, is continuously shedding some skin cell when they are no more life in it, and it become dead, and it not become the part of the body, and then it subsequently get detached from the body and start coming in surrounding environment. We have observed, which we will be talking throughout this presentation, Ronnie, that often this is overlooked, as I said that in my introductory remarks, while we investigate the indoor contaminant, but it is omnipresent, and I'm pretty sure most of the time this contaminants are going to be there if you are going to evaluate the indoor environment. So in nutshell, what we are going to do during this seminar, we are going to focus some of the facts about the skin cell and how it interfere or how it influence the overall health and hygiene of a building. That's our intent.
Okay. Sounds good, Dr. Sahai. Why is it important to pay attention to these particles when talking about building health?
Well, there is two things. One is the human health. The other aspect is the building health. So let's take the topic one by one. The building health is going to impact the quality of living. If there is a problem in and around a building in terms of environment or in terms of perpetuating flora, fauna, or any chemical or any kind of pollutant that may influence or impact the quality of life. So it deemed necessary to understand that what could be those causal element or what could be those element which can influence the health of a building. So obviously skin cell is also identified as a source of those influencing material. Why? Because it provides a nutrition source to the microflora. When I say the microflora, that include various kind of microbes, including but not limited to viruses, bacteria, mold, and others. And we have also observed that about 75 to 90% of the house dust is made of a skin cell. Isn't alarming that if you are making a survey, you will find that out of 100 particles, 75 to 90 particles are human skin cell and appears as an epithelial cell, and it stains beautifully if you look under the light microscopy or under a microscope. It appears very distinct and you can easily identify. Actually, it is one of the easily identifiable microflora from the dust. According to an estimate, about 500 million cells per day one human being sheds. It means that about, in terms of mass, 30 to 90 milligram of skin flanks drop off every hour by every individual. Isn't alarming? That's why I was telling that it is most commonly observed contaminant of indoor environment, but most ignored one. The other functionality of this skin cell is support the growth of the microorganisms, not only by providing nutrition like I was telling, but also providing a platform so that the microbes kind of associate themselves with that and keep growing. So maybe those microbes are growing on the skin cell, but you can see their effect on the human body through the building health, because they are going to perpetuate in the surrounding of that environment, and the occupants are going to realize in terms of symptom when they are going to react against this particular contaminant. That's why I said that it is a very, very influential to health and hygiene, especially those having a compromised immunity. But I want to make sure I clear this point that at this time there is no big medical correlation available. There is a lot of undiagnosed symptoms, and we believe that some of the symptoms are directly or indirectly can be related with the human skin cell. But again, I underline that at this time there is no definite medical correlation, but it has been observed through our surveys in the indoor environment that if the skin cell is in the copious amount, then we have definitely get some complaint by the occupant in terms of their health.
All right. We are going to be having questions throughout this webinar as well, and I'm going to go ahead and start the first one. As shown on our invitation to this webinar, if you get 4 out of 5 questions correct, we will be sending out $10 Starbucks gift cards. So I'm going to go ahead and start with this very first question. Where are dead skin cells commonly found? House dust, kitchen stove, all of the above, or none of the above? And go ahead and give about another 10 seconds for it. And it's going to be closed now. Just to share the results with everyone, we got 43% getting the correct answer. It is house dust. All right. So let's go ahead and continue. Don't worry if you got it incorrect this round. We still have 4 more chances. So let's go ahead and continue. All right, Dr. Sahai. So now we know a little bit more about the skin cells and what they are, but can you tell us about some common risk factors that are associated with this aerobiota?
Yes, sure. The common risk factor associated with this particulates of indoor environment is allergenic reaction, because as I was explaining earlier that there are several allergenic reaction or symptoms are unexplained, and authors believe that some of them may be due to directly or indirectly influenced by these biota. As I earlier said, that this can harbor various kind of microbes or other chemical for that matter that might be allergenic to susceptible individual. One thing I guess many of our viewer have experienced in past is that odor complaint and comfort issue while they are in air conditioned building. And what they have noticed, and at least some of them has shared with me in case of my routine laboratory practices, that they get some very weird smell and they cannot explain that how this smell is. In absence of that, some of the occupants has shared with me that the odor is very persistent and it looks like a dirty socks. So sometimes it is called as dirty sock syndrome. And it is very uncomfortable because you don't want to be around a odor which is bothering you. So that is one of the very commonly observed risk, which is directly related with the comfortness of living. One thing I want to emphasize in terms of risk, that it impaired the functionality of HVAC. And let me elaborate this particular topic a little bit more, because heating, ventilation, and air conditioning system is the one of the essential component of modern building, because air is being artificially managed. We have not like a legacy of interchanging the fresh air by cross ventilation, rather it is being managed through many a time a central system where the fresh intake is limited. But what happened in due course of time, these skin cells are going to settle down throughout the HVAC system, especially on the coil and the surfaces of coil that restrict the airflow. So we have two kind of impact in terms of energy, because we are going to waste energy because the constriction and the flow of air go down. And on the other side, it is become an incubator for various microbes such as bacteria, mold, etc. And that has directly linked with the health, or the occupant who are there is going to get exposed, and if they are susceptible, they are going to show the symptoms, or if it is a disease causal or the pathogenic organism that is going to cause disease in the human body. Skin cells are also an indicative of mite infestation, because the mites love skin, because actually they live on skin cells, and the mite is also directly linked with the indoor allergen, because it is a type of allergen which is very commonly reported from indoor environment. So overall, there is a greater risk factor associated with that, which influence health and hygiene.
So there's a lot of risk, and it is pretty harmful, huh?
Yes, it is very, very harmful, but you should not forget that this also helps sometime to reducing the indoor pollutant. You will be thinking that I'm going crazy. A minute ago, I was telling that it influences health and hygiene, but now I start saying that it reduces the indoor pollutant. How come it is possible? Well, first, I have explained you that what it can do if it is in uncontrolled manner in some indoor environment. But if it is not in that uncontrolled fashion, then there is certain chemicals or certain essential oil, which is obviously in every human body, is very helpful in reducing the level of ozone, which is about 15% reduction in ozone. And ozone is one of the substance identified as a cancer causing element in the indoor environment. So this is one of the beneficial aspect. If we have the skin cell, they can reduce the ozone in indoor environment. But again, the question arises, how good is good, how bad is bad? So we will be also discussing and looking some data that how we can see that this skin cell is a problematic or this is going to be a friendly approach. The other important factor which I again want to bring here is that it provides the nutrition to the growing indoor microbes, and that's how it help microbes to grow. And those microbes many times is helpful in recycling the material. Again, question arises, what kind of microbe we are talking about and what kind of scenario we are looking. Then it also indicate the occupancy load in terms of forensics, forensic science, when we are trying to figure it out that what is the occupancy, or how much people, or what is the adequacy of the occupancy, and how many people at a given time in a building. This can be a very good surrogate to indicate that how many people are there, because simply the more people, more skin cell. And then it also indicate the dermal health of the occupant in those situations. So it has also some beneficial aspect when we talk about the forensic science.
It's a lot of good information, Dr. Sahai. Can you talk to us about how you can collect and identify these bioparticulates from building environments?
Yes, sure. This is one of the very integral and essential component of our presentation, and that's how we have collected the data. The skin cell can be collected from the surface or from air by using different method. From the surface, we can collect it by using the BioScan 400, and we can use this BioScan 400 strip to touch simply take that strip and touch the location where you want to collect the sample for the skin cell. Other method is environmental bulk sampling method. Simply you collect the dust or debris from a place by using filtration techniques such as using a dust sock, etc. And then you send the bulk sample to the laboratory. Laboratory make the preparation and prepare the sample for that. However, the air sample is collected with a technique called as a spore trap method. In this method, there are several commercial media available, such as aerosol, microfiber, etc. And you can draw certain volume of air through these media, and then you can submit the media to the laboratory, and they can do the analysis. Mostly the examination of this skin cell in terms of indoor environment can be as simple as with microscopy such as bright field, etc. However, electron microscopy can also be performed to know the detail. But on routine basis, the microscopy through bright field is very useful, and it gives a very good idea about the concentration and the other nature of the skin cell.
All right, that's good to know, Dr. Sahai. We're going to go ahead with our second question now, and I'm going to go ahead and launch it for everyone to see. Which risks are associated with dead skin cells? Mite infestation, odor and comfort issues, HVAC functionality, or all of the above? Go ahead and give everyone about another 10 seconds. And I'm going to go ahead and close the poll. Let's see how everyone did. We got 86% getting it correct. It was all of the above. All three of those things can be associated with dead skin cells in an indoor environment. So it's interesting to, we talked about how to collect the samples, Dr. Sahai, and then you explained how these samples can get examined. Especially here in our laboratory, we do the microscopy typically, and it's really interesting to see these particles under the microscope, right?
Yes, see those bright blue particles? They are the common skin cell taken from the surface. And this appears very bright, as you can see, and it is very distinct. If you compare with a fiber or other kind of opaque particle, it is very distinct. In the next slide, I will show you that you can make some assumptions with the skin particle itself. In the next slide, this slide you can see I'm trying to explain here that when the healthy skin is set, then it is going to be a long, little larger particle. Whenever the skin cells start getting further deteriorated, then will start getting a smaller and smaller. As you can see here on this particular one is a very long skin cell. The size is given here. It is about 84.85 micron, which is kind of very, very large in terms of microscopy. In the next slide, I will also show you some of the other measurement which can give you a very good idea. And I know that many of you are not very technical on that, but I have taken a surface area, and the surface area of this particular skin cell, which is right here, is about looks like over 100 or approximately 100 micrometer square, which is a big surface area. And on this area, a lot of microbes are accommodated, and especially it is a bacteria such as staphylococcus or other human commensal bacteria can be discharged in the environment, and they can be very cozy with the available nutrition in this skin cell, and they can be just in the indoor environment. And once it starts getting further deteriorated, that can set free in the ambient air or on the surface. And if you get infected with that, then you will be obviously start showing the symptom. The next slide is also going to show you some more skin cell that it is a lot of variations. It can go very simple and very, very small, as you can see in this one maybe below 1 micron, but some of them are going to be very big, over 100 micron. So there is lot of variation. So far the shape is concerned, there is no consistency in the shape, and I would say that it is sometimes pentagonal, hexagonal, oval, and other sizes, and the shapes are very prudent and is very commonly encountered, especially when these particles are examined under the microscope. The next one is going to be the one which is going to be comparing the uh, surface on the skin cell, which I was showing you earlier was collected from air, but on the surface also there is no specific shape of these skin cell, but they are various shade, and they are various size, and then again they contains a lot of nutrition that can support the growth of microorganism for a while. The next slide is going to give you some more idea about the how small or how big the skin cells are. This particular slide, which is going right now on your screen, is showing you a very beautiful creature. I'm not sure how many of you have guessed that or not, but this is a dust mite that is known allergen. And as I was telling earlier during my intro, that these dust mite feed on the skin cell, and that can be very, very good for them, and they can sustain themselves just feeding on skin cell for a while. And nevertheless, they are the decomposer, I mean the dust mite, but at the same time it also harmful, because on the one hand it is recycling the skin cell, but on the other hand it is very, very potential, or having potential to create allergenic symptom in occupants.
All right, Dr. Sahai, those are some really nice pictures to kind of show everyone an idea of what skin cells can look like in their sizes. And, just kind of going back to that fact that you told us at the beginning that humans shed 500 million cells a day, even though they're so small, that's such a large number that you can see how that can easily become a problem, day after day in an environment. We're also going to go ahead and launch our third question now. What can the dead skin cells of dust microflora reduce? Hydrogen, oxygen, ozone, or chlorine? Go ahead and give everyone about another 10 seconds to get their answers in. All right, I'm going to go ahead and close it. And we can see that 50% got that one correct. It was ozone. So now we'll go ahead and continue with our presentation. Dr. Sahai, can you elaborate your experience with these particles in building environments?
Sure. We have had experience of collecting samples from various places, both from commercial and residential facilities. And I can share some of my finding with the surface particulate from the indoor environment in terms of skin cell. This particular table summarizes my finding of observation about over 14,000 samples. They are collected from the surface, as I explained. And you will notice that the skin cell fragment in all the category, which I have categorized for sake of convenience to explain my participant, that skin cell is one of the very predominantly recorded contaminant, in lack of better word, in the indoor environments. The number, let's look the number. Out of 14,056 sample, we have recorded about 13,073 sample positive and about only 983 sample negative. The lowest concentration of skin cell per centimeter square is about 4, and the highest is 102,500. The median value of this particles is 160. The mean value is 949, and the 67th percentile is about 356. So what it means that about one third of the building contains or reflects 356 skin cell per centimeter square, and that number is obviously the third largest number when I compare it with the other category which I have put for the opaque particle. For those who are wanted to or interested in knowing what those opaque particles are simple dust and debris which appears very opaque under the microscope. So we have counted all those particles, and out of 14,056 there is 13,831 particles are like positive in terms of like the opaque particle. Only 225 samples are negative for the opaque particle, and again the highest concentration is 447,000, which is a lot of particles, and the median value is 1400. Mean is about 4,727, and this 67th percentile is indicating 2,883. What it means that more than 60% building is going to have about 2,800 or a little bit over 800, to be accurate, 2,883 particles of the opaque nature. Then insect biodetritus is the other category where we have recorded about 2,200 sample uh, positive, and only about concentration 5 mean value. So this is not very uh, active or very dominant, but I wanted to just give a real quick at a glance view on the fungal spores, which is about 8,816 samples are recorded positive, and then 5,240 samples are recorded negative. The mean value of this is 6,239. So the fungal spores are one of the very predominant biota in the indoor environment. However, that 67th percentile concentration is indicating only 32 spore per centimeter square, whereas others, which include various kind of particles such as black particles, brown particle, meaning the rust particle, etc., and their concentration is over 1,452 on an average basis, and 67th percentile indicates that it is about 500 in number in terms of count per centimeter square. So that was at a glance view for the surface sample. I'm going to show you next slide for the uh, value in air. So
And real quick, Dr. Sai, just to interrupt you, we did publish a paper in the Continental Automated Building Association's magazine that does also contain a lot of this information, and I've actually attached that file as a handout through the GoToWebinar. So in case you need to look back on this or you want to look a little bit more into it, we do have that available as a download as well. Go ahead, Dr. Sahai.
The same statistics I want to give our viewer for the air when we are exploring the skin cell in the air. The result is very interesting because I have about 5,197 sample for various particle, skin cell, opaque particle, uh, insect, fiber, pollen, fungal, and others. And I have noticed that out of this 500, 197, the positive sample is 5,156, which is a very large number because only 41 samples were negative or devoid of skin cell. The lowest concentration is about 22 particle per cubic liter volume of the air, and the on an average concentration, meaning the mean value is over 4,883, and 67th percentile value is indicating 4,940. So this is one of the very predominant particle in the building Let me compare real quick with the fungal spore. The fungal spore, you see that only 4,615 sample is positive and about 582 samples are recorded negative. But the value of the skin cell on an average is much lesser in terms of skin cell. It is 3,573. And that what we can say that skin cell is more predominantly floating in our air than even a fungal spore, and the 67th percentile is only 488 counts per cubic liter volume of the air or meter volume of the air, which is almost 1/10 of the skin cell. So skin cell is very predominantly floating contaminant of indoor environment. Now, I wanted to give the graphic representation. As you can see, in terms of airborne particle, the skin cell, both in the airborne situation, in terms of total number of sample, almost every sample has shown the skin cell. The insect biodetritus is the one where we have very smaller set of sample has shown a positive correlation or positive sample in terms of total sample. But you can see that the skin cell and the opaque particle in and along with the others are neck to neck. I want to also show the next graph, which is based on the surface uh, particulate. Here we also see in that the skin cell, both in the total number of sample, if I compare, then it is almost neck to neck, because almost all the sample has exhibited the presence of the skin cell minus a few, and so does with the opaque particle and others, whereas the fiber and the fungal spores are there, but not nowhere near than the skin cell. And the insect biodetritus again is the least recorded biota.
So looking at all information, it looks like it is definitely one of the predominant contaminants of built environments.
Yes, and then this average concentration in terms of the pie chart, you can see that the skin cell, which is indicated here in the light blue for a slice of pie, if you want to compare in that way, is really after opaque particle and the others biota, the skin cell is really the dominant biota in air. And in terms of surface, you can see this pie chart. It will be coming in a minute. You can see again the skin cell is one of the very predominant or top three predominant biota in terms of number.
Actually, before we continue, we're going to go ahead and launch the next question. Which method can be used to collect dead skin cells from the ambient air? Spore trap, Bioscan 400, swab, or none of the above? We'll go ahead and give everyone another 10 seconds. And I'll go ahead and close it. And it looks like 67% got it correct. It was the spore trap method that is used to identify skin cells from the air. So, Dr. Sahai, in your opinion, what are the important things we should learn about this particulate?
Like I said, the skin cell is one of the common constituents of dust flora of indoor environment, and it typically come as a flanks of skin from the dweller skin mostly. Then the dead skin cell in indoor environment has both negative and positive impact. I have explained these two phenomena, that sometime it is helpful in reducing the ozone concentration, but many a time it is having a negative impact because it provide like nutrition, and it create like nuisance and odor and comfort issue. The higher concentration of these particles in HVAC system in comparison to the other places while investigating indoor environment, and that make me conclude that the accumulation of skin cell and the bacterial degradation of the skin sequential in AHU and the associated ductwork can cause a urine-like smell in air conditioning system, and it is identified as a potential source of building contaminants that can be issue of like odor and uncomfortness. The elevated concentration within the heating, ventilation, air conditioning system may be nuisance, odor causing, and problematic from the health and hygiene point of view, and that really not like impact our living or the building health, but it also like have a greater interference on the total energy cost, because the airflow is restricted and it requires more energy to maintain the same system. So that sort of like main or focal point we need to concentrate. As I said earlier, that at this time there is no perfect medical correlation, but definitely the health and comfort issue is associated with it, as we have find in our survey with the occupants that they have like some uncomfortness while they are occupying a building with a higher concentration of the skin cell.
Could you share some of the important techniques to alleviate this contaminant from building environments, since you're saying it can cause some problems for people?
Yes, sure, because this is one of the very commonly asked question, that how I go about like taking care of these particles. Well, very simple technique, and I guess many of you are already aware. I'm just brushing it, that a good housekeeping practices on consistent and periodical basis, that I want to underscore. On periodical basis, we are very good at sometime cleaning our house. We do good cleaning, but when we are in mood, but we forget like next like several days, and that should not be the case, because it should be on the periodical basis. That makes sense.
Yes.
Then the topical cleaning is the other mechanism by which it can be alleviated. Use of eco-friendly disinfectant to clean high touch or frequently used surfaces is helpful to reduce the concentration of skin cell. The filtration equipment of HVAC system with a good MERV rated filter is suggested, so that these particles should not be in terms of infiltration or in terms of like filtering these particles. If you have a good MERV rated particle, it is going to be arrest right there in the filtration system, and you should not be dealing with this problem. The HVAC use green technologies such as steam cleaning process to clean the evaporator coil, blower case assembly, drain pan, and other component of the HVAC in terms of making the airflow or enhance airflow and maintain the system life as a whole, and you can save energy. So definitely one of the aspect is to look at your HVAC for a regular maintenance, and use such as a steam, as I said, like some eco-friendly technique to clean it in order to stabilize and optimize the system as a whole.
All right. And before we go ahead and conclude this webinar, we will ask our final question. Dead skin cells can impact what? Health and hygiene, HVAC functionality, all of the above, or none of the above? Give everyone about another 10 seconds to answer. And I'm going to go ahead and close the poll. And it looks like 63% of you got it correct. It is all of the above. Let's go ahead and close that. And now just some final words for joining our webinar. We did have one question, Dr. Sahai. I think you did touch on it briefly, but maybe you can elaborate a little bit more with it. do you find viruses and bacteria catching rides on the skin cells?
Yes, that's a very good question, and yes, they ride on that, and not only that, they can just survive. As I said, that they also is the source of nutrition, so they can survive just like feeding on those cells for a while before they get launched in the indoor environment.
All right. Thank you for that, Dr. Sahai. We are just about out of time here. Go ahead and let you all know that we will be having a booth at the AHR Expo in Atlanta in January 14th to 16th in 2019. You can visit us at booth B965. If you have any further questions for us on this topic or anything like that, I have the contact information up here for you. Feel free to talk with us, send us a question, or just talk with us about any topics that you may want to see or have discussed in the future. We're always very happy to talk with people and share our information. We'd like to thank everybody for coming and listening to us talk about this important topic, human skin cells, and appreciate you being here. Thank you.
Thank you again for every participant of ours, and we always look for a topic of interest among the participants. And as Ronnie said, that if you have any question, these webinars are like sort of like information sharing basis. They are free of cost to our viewers, but we are always open to discuss it further. So if you do not have a chance, or you did not have a chance to ask the question, but if you do have one, please do not hesitate to email me at my address rsahai@pureaircontrols.com, which is given on the slide. I would request Ronnie, let this slide for another couple of minutes here so that our viewer can write it down, our address. Other than that, if you have any topic of interest, you can always send us, and we will be promptly considering that to include those topics in our future webinar. Again, thank you very much, and thank you for participating today's webinar.
Thank you.